Good morning, everyone. Welcome to our program, OmicsLogic Metagenomics. Today, we'll be talking about the DADA2 pipeline. And so before we start talking about the pipeline, I want to give a brief review of what we've covered in the last session and kind of talk a little bit about the program itself and where are we going. So first of all, the program is built for us to be able to understand the role of microbiome in the context of data. And so we started talking several sessions ago how metagenomic sequencing has really transformed our understanding of microbial communities, and the understanding of the different types of metagenomic sequence generation and analysis can ultimately help us understand the microbiome, understand how the microbial communities live and function together, and also how they impact the different aspects of the world that we live in, including agriculture, organ function, and general health and well-being for all of us. So it's an exciting and important topic, and I hope that today we'll get some practical hands-on experience learning about this pipeline. In our last session, we talked about mapping on databases. So whenever we have any kind of sequencing, either the full 16S gene, 16S ribosomal RNA gene, or when we look at the hypervariable regions, like what we'll do today, in either case, we rely on these databases. And the quality of databases, the aspect of what kind of information they contain, how is this information organized to represent the taxonomy, for example, the Open Tree of Life taxonomy, and also how manually curated the information is going to play a big role in how we will be able to interpret the results of our analysis. So we talked particularly about the Silva database that has manually managed a collection of data and reviewed and validated about bacteria, archaea, and eukarya. So we talked about that last time, and then we even mapped some data from the human gut microbiome project on this reference database. And so what we did is we used the Bowtie2 alignment algorithm that was mapping the data that we have from sequencing of that of one sample, and we can generate a sort of like a table of abundance where we can see for each sample. Last time we did one sample, but you can have multiple samples, and we talked about where to get that data. We can map it on the database and then annotate the results by taxonomic order of different species, phyla, and genomes that we find. So we generated this table of abundance, and we talked briefly about what would that mean, right? And how we can look at the abundance numbers, we can filter them, and we can ultimately find the most abundant species or phyla that we find in the data. Now, we promised several times already that we will have Metagenomics 2 available soon. And so I'm excited to announce that Metagenomics 2 is available already. And it's a part of the Metagenomics collection. So we now have Introduction to Metagenomics, Metagenomics 1, and Metagenomics 2. And so the Metagenomics 2 course is going to be essentially what we are covering today in greater detail, and also will provide some additional information on some of the things that we just cannot fit in a single session like this. But if all of you can review some of the data here. Sorry about this. Let me just turn off my notifications. If you all can take a look at the collection, let's just briefly take a look at some of the topics that are covered. So you will see Metagenomics 2 right here. And I think what we've seen in the past is that anyone that takes these courses online will benefit greatly from the sessions that we have, because you have more opportunity to stop and think about some of the aspects of the practical hands-on exercises that are described here. And you will see the step-by-step guide and also the preparation of the metagenomics analysis pipeline. So those are some of the things that we'll cover today. Okay, so in our OmicsLogic Metagenomics program page, which is this link, you can see all of the activity from the participants, and you can see that a lot of the people are starting to work on the practical side of the assignments. And I think that's very important, because ultimately reading about it and then applying the understanding to a real data set is ultimately what gives you the real understanding of what it is that we're trying to do. So I would encourage everybody to not just complete Metagenomics 1, but also try the practical assignment. You all have an educational account, which means that you can use the links or use an NCBI run table on the platform. And if you have any questions about that, we'll be happy to answer your questions. So you can reach out to us by email, on the chat, or during the session. I'd be happy to answer any other questions that you might have as well. So today we want to continue and explore the DADA2 pipeline. And the DADA2 pipeline really was designed to address several of the challenges related to analysis of data, where we have the Amplicon 16S gene, but only the hypervariable regions. So let's review again, what are these regions and what are some of the challenges that arise from sequencing just that data. As you remember, the 16S ribosomal RNA that looks like this has a lot of information that is shared across all of the bacterial species, so all of the eukaryotes. And it also has some of these hypervariable regions. So the full sequence of the gene that encodes for the 16S ribosomal RNA is approximately 1,500 base pairs long, but the conserved regions are fairly small. So they're between 30 and 100 base pairs long. And so if we want to sequence with high depth just specific regions, and those regions provide us the information about the taxonomy of the bacteria that we have in our sample, then we can really utilize to the fullest the sequencing capability of, for example, Illumina MySeq. So Illumina MySeq, the reads are about 250 to 300 base pairs long, and putting those hypervariable regions, one or two hypervariable regions in a single run, enables us to get some high coverage of just those regions. The hypervariable region number 4, for example, so you can see it right here, can be amplified using these specific primers. So these are going to be used in the library prep, and you can see that we will, as a result, we will get these 288 base pairs to 290 base pair long amplicon covering the V4 region. So in a lot of times when you read through the method of the data preparation, you can also take a look at what is the region that is being used. Sometimes it's going to be V4, sometimes it's going to be V3 and V4, and so you can then get different level of detail from the sequencing that you ultimately have. The meaning of the hypervariable region, so you can see here, this is mean frequency or the most common residue in 50 base window, right? So if you think of frequency, how often a single nucleotide changes, you can see that the variable regions are right here. V4 is right here, V3 is right here, and this is the position on the 16S ribosomal RNA gene. So you can see that the lower the number right here, the less conserved they are. The higher, the more conserved. Meaning that if you were just to sequence these regions that are at the top, you would essentially not be able to distinguish at all between the different representatives of your sample. And the lower it goes, the more you can actually see. But also look at the length. Length is also important. So you can see that V3 and V4 are some of the longer ones. And V9 obviously is long, but because it's at the end, you're not going to get such good information from that. So you can see that sometimes different combinations of these regions are going to be used in sequencing. Now, when those are sequenced, those paired end reads are going to be used to create what's called organizational taxonomic or operational taxonomic units. So the forward and reverse reads, by aligning them together, we are going to get really good coverage. So we have high confidence of the actual sequence. But because we only have the taxonomic units, OTUs, we're actually not looking at the full genome, and we're not looking at the full 16S ribosomal RNA. We're looking at a unit, at a portion. And so, or how do we get them? Well, so what they represent are actually groups of closely related individuals, right? They don't represent an individual, they represent a group. And typically those groups are done using clustering. So for example, you can use hierarchical clustering to combine them together. And OTUs will be clusters of similar sequence variants of the 16S ribosomal DNA marker gene sequence. So the number of known OTUs can be inflated due to sequencing errors. So for example, if you look over here on this side, if they all contain a similar variant from some reference, we will say that, okay, these all belong to the same group. And so that's how we are organizing our information after sequencing, right? So, and this is one of the challenges that we're going to be facing. As you remember, standard pipeline, we're just mapping it on database. And when we're just mapping on the full sequences in the database, really, they can fall in a lot of different places. And here we're trying to reduce this kind of diversity that is not true, right? We're really trying to get to the bottom of, well, how many of each kind do we have? Now, to understand how this can be applied to a real project and what the data will actually provide us with, we can take a look at the project that we have prepared for today. And so the project is the gut-brain axis. It's related to how the microbiome affects behavior. And so we're going to look at mice that were fed different diets. I think there is a separate PDF that is in one of the links that was pasted with the reference publications, where you can see just an overview of the gut-brain axis. Now, there are many different ways that the gut and the brain can be interacting, but I think in this context, what they did, they gave high-fat diet, so some mice at a young age, when the microbiome is forming, so at a younger age, for example, for humans, the microbiome typically is formed by around age 4 or 5. And there's an interesting study of twins that was done in the UK Twin Project, where they looked at the microbiome, and the microbiome could be different between these twins. And so the question is why, right? So if they were born from the same mother, if they were kind of fed the same diet from an early age, lived in the same house, why would their microbiome be different? And really, there's not yet a clear explanation to that. It could be a number of different factors, including different infections that they acquired during the early ages. It could be the different treatments that they received. But ultimately, what we know is that the microbiome could change even between individuals that have very similar genetics and very similar environment. And also, we know that they are stabilized, that microbiome will be stabilized by age 4 or 5 in humans. In mice, it's significantly earlier. So what they did was they had young mice. The mice were fed with a specific high-fat diet or a regular chow diet. And so then the mice that were given the high-fat diet microbiota they were observed to have significant and disruptions, exploratory, cognitive, and stereotypical behavior compared with mice with a controlled diet microbiota in the absence of significant differences in body weight, right? So we're not talking about changes to the weight, changes to the diet. We're just talking about kind of implementing a different type of a microbiome. So this phylogenetic analysis also that we'll look at today will help us identify some distinct microbiota that is differential between groups, and how that affects things like alpha and beta diversity, taxonomic distribution, and also metabolically active taxa, right? So we know how functionally some of the taxa works, and so we can also look at that. So I think what is going to be interesting is for us to look at this project. We're going to look at just a small subset to just understand how the pipeline works. But then you're, of course, welcome to take the full data set and play around with it and see what's in it. And so I'll walk through some of the preparation of data and then how to run the pipeline. What are the steps of the pipeline? What are some of the outputs of the pipeline? And then you'll be able to kind of try and replicate the same thing or even try and work on a different data set similar to this. So again, what is the data? The data is we have 8-week-old males. They live for 10 weeks, one group on high-fat diet, one group on regular chow. First of all, their samples are then taken and sequenced, and their microbiota is transplanted into recipients. So you can see here that some 3-month-old males, that is older males that have already an established microbiome, they were treated with antibiotics to reduce their bacterial load. So really their microbiome was simplified. So you can see, for example, after antibiotics, a lot of the diversity is reduced significantly. Then they had implant of that high-fat diet or the regular chow microbiome from the donors. And then they did some behavioral testing, right? So ultimately, for us to understand what changed at the level of microbiome, we need to analyze the sequencing data. When we need to see how the microbiome affected their behavior or their condition, psychological condition, we can do behavioral testing. But also, there were some samples taken after the fact, right? So now let's talk about the analysis itself. So before we jump into the pipeline itself, let's take a look at the data that was provided to you. So we can go to the data. And if you haven't downloaded the data, please download. We have the data there in the chat box. Here we have the files. And so we essentially have the full data set right here in Excel. So let's just take a look at what kind of samples do we have. So you can see that for each one of the sample IDs, we have the recipients, right? And then we also have, this column shows us the cecal and the fecal samples. So what is the difference? Fecal is, of course, the fecal matter. Cecal are essentially a portion of the gut. And so what we, as an example, what we can try and do is compare from just fecal samples, right? We can compare high-fat diet versus chow, or we can take something like fecal versus cecal, right? Just to understand whether there's going to be a difference. And as you remember, 2 sessions ago, we kind of talked about the gut microbiome and where is that microbiome. And we talked about some of the challenges with analyzing microbiome data. For example, why fecal samples would be different from cecal and things like that. So you can also see here that each one of our samples is going to have 2 types of data. So it will be paired and read. So you see at the end of each file, you have _1 and _2. So now let's take a look over here. We have a prepared SVL file, which is a reference file. And the reference file will contain just those links that we saw, right? So over here, you can see some of those links, right? You can see the number of the sample, and you'll be able to find that sample right here. So for example, you can see we have BKCM1. So BKCM1 is probably, yeah, this one. So then you also need another file, and this other file is right here, the supplementary table. And the supplementary table is actually done for further visualization of the data that we'll talk about here in a little bit, so that we know what is essentially the difference, right? So what is the, so whenever you prepare your pipeline, you have to prepare the data in such a way. You have a text file that tells the pipeline something about your files, and then you have the links to the files themselves, which are going to include at least 2 different, files, because this is paired and sequencing. So let's prepare the data that we would like to analyze. I think it would make sense for us to take this kind of a data set that helps us compare chow versus high-fat diet recipients. How do we prepare an SVL file for just these files? Well, as you can see here, we have a lot of them, right? So why don't, and also you can see that the F at the end stands for fecal. So we need BKF01, right? So that's right here, 1 and 2. BKF01, that's this one. Then we have BKF2, so that's this one. 3. And then 13, 14, and 15. So essentially what we can do is remove all of this, and these are going to be cecal files. You see that they're C instead of F, so they're cecal files. So what we can do is we can just take, let's just take this new data set. Okay, so essentially we have 3 of each, 3 of the high-fat diet and 3 of the regular diet. We can copy this and just create a new text file, place it in here. Make sure that you have plain text, so you don't need any additional information, right? And then you can save it. We can save it. Let's give it a name. So let's go to the same folder. Again, I just want everybody to understand the process, so we'll be doing this together, because in our next session, we will actually be working with your independent projects, right? And so this process is going to be important. So this is high-fat diet versus chow, and this is just fecal. You can save it, and then you can just change the name to SVL. So now we have this file, and the names should match exactly what we have in here. Okay, so these names should be exactly the same. Now let's take a look briefly at this example. We're not going to run a demo pipeline like we did last time. We're actually going to prepare the full one. And this pipeline has two sections. So you can see the data to section, and also you see here phyloseq. Both of these are essentially R libraries that are organized in a way that they can work together. And what they will produce are some results in text format, so a table, but also some visual formats so that we can take a look at a summary of what the data really shows. So let's go to the platform. Server t-bio.info. And the pipeline is going to be found right here. If you just want to play around with this data, you can click on this demo. But now we're going to go inside. So please stop me if you kind of have any questions about what I'm doing right now, or you would like me to pause until you kind of catch up. So please just put that in chat. Okay. So here you can see that you do have an option to upload your own database. The database that is uploaded is going to be right here, just Silva, but you can also upload your own database. So if you want to use Green Genes or anything else, you're able to upload that yourself. But we're going to just use local, the Silva database. Now tell me, do you think this is paired end or single end reads? Exactly, yeah, paired end, right? We talked about this kind of sequencing, so in fact, that's the only option. And are these FASTA files or FASTQ files? FASTQ, thank you. So FASTQ files, we selected the Silva database, we already know what that is, and now we need to upload the data. So here, instead of using the raw files, what we'll do is we'll upload the bulk upload, URL bulk upload. So you click on this, and then you have to select from the folder that we have. Okay, and let's use this SVL that we just created. Okay, if you want me to pause or explain anything, I'm happy to do so. Please let me know in chat. So here you can see supplementary text file. So that's the file that we just created, that text file. So we'll just add files. And it's going to be called supplementary table. So we'll choose that. And then submit. Okay. Now, as usual, we click on start. We can ignore this first pop-up. You can see that we have the option of pre-processing. Pre-processing will do trim-o-matic. Trim-o-matic reduces those primers. In this case, it's not going to be relevant because we only selected for those hypervariable regions already. So we're not going to have that problem. And now we can just focus on the data to pipeline. So we can first click on the filtering, and you can see that we have some inputs here. I don't recommend changing anything, but you can see what types of inputs you will have. So first of all, the read length, right? We know it's going to be between 250 and 300. And what filtering does is it's actually going to look at the error rate in read quality, and it's going to trim that by a certain length where there's minimal number of errors. Okay, so it's going to be important, and by having some experience working with these files and looking at the outputs, you will see how this actually plays a role. So let's leave it here. And by the way, what I also recommend in parallel is let's just look at the demo pipeline where you will be able to see some of the outputs already. So here, if we click on Start, and then we look at QC filtering, you can see this image right here. And this is essentially what filter does. So you can see that the quality of the read, this is the length from 0 to 300. And you can see that the quality starts going down at this point, right? And starts going dramatically down. So this is the portion that we would like to trim, okay? And sometimes you can see over here, for example, the quality here is pretty bad, right? So that's what we're looking at. And we're looking at also both types of read. So that's QC filtering. Now here we have replication counts. What is replication counts? So replication counts, we have a lot of reads, right? And so when they're trimmed and they're kind of, fairly reliable sequences, we need to eliminate those reads that do not have a matched pair. And so we're not looking at abundance like we did in last time. We're looking at taxonomy. So to look at taxonomy, we're actually looking at repetitive information as a single point of information, single data point. So we don't want all of the reads and the number of reads. We just want to combine the ones that have been denoised, right? They're merged, and then we want to count them as operational taxonomic units, OTUs. And then we will combine them together in these groups by similarity, right? So here you can see that we will do pair merge. So here, for example, if we go to the next step, pair merge, you will again have some input, right? So how much overlap do we want our reads to have? So you see that the minimum overlap here is 12. Out of 270, which is the number that we kind of trimmed it down to, we can have a minimum overlap of 12. We can increase this overlap if we want to improve the quality, and also maximum match and overlap. If we have an overlap of 12, how many individual nucleotides do we want to be different, right? So this is very flexible, actually, right? These settings right here. And now we need to combine them into these operational taxonomic units, which will be done using clustering. So we actually want to look at similarity of sequences and combine them together. So if we go and click here on OK, you can look at OTUs, right? As you remember, we are going to combine them together by similarity of sequence. Let's click on OK. Now, what other problems can happen? Other problems are what's called chimera. Chimera are reads that have the beginning from one position and the end, or like the second half, from a completely another position. So for example, you might have something that comes from one gene or one hypervariable region and then another, but they're combined together. And when we denoise the data like we did before, now identifying those chimeras is going to be easier, and we can improve the quality of our organizational taxonomic units. When you just map on the database, you might have in your results a lot of chimeras, and then you're thinking, oh, maybe this is some new species that is just not annotated. How do we interpret that, right? And that actually adds noise to your data. But in this case, we don't want to look for abundance. We're looking for kind of diversity, and that will help us to remove the chimeric sequences, which could be, anywhere between, you can see here, makes up about 21% of merge sequence variance. So this is a lot. 20% of your data could be completely misleading and false, right? And so after you do this step, you get it down to about 4%, so which is a really nice improvement. Okay, after we do this, we need to assign the taxon. And so we know how these specific hypervariable regions are annotated in the database, right? So let's click on Save Here, Assign Taxon. What it will do is now, based on these filtered out, cleared up, merged, and cleaned up from Chimera's reads, are now OTUs, operational taxonomic units. And now we can look at where do they fall in the taxonomy, right? Which is what we want to calculate diversity, because taxonomy ultimately shows us not just this belongs to this species or group, but it actually has a certain relationship, right? And that relationship is measured in evolutionary distance. And that's where we come to phylogeny, right? So we have phyloseq right here. We have first have to prepare the data, and then here we can select how we can use that taxonomic relationship. So here you can see that the taxonomic level goes all the way to kingdom. Of course, the lower you go, the more kind of diversity you expect. So you can decide which one you want to use. We know that the Silva database typically goes to the genus level. Some will go to the species level, right? So we looked at that table last time, and we saw that a lot of the annotation will just be not available. Below the species, definitely, but a lot of times also below the genus level, right? So if we want to get all the information that this kind of sequencing provides us, and different types of hypervariable regions provide us different level of taxonomy, we can map it appropriately. So let's select here genus. You can see that it will calculate abundance, and also it will show us some of the information in a colored way, right? So here we can actually change this, and we have different ways of calculating the ordination. And we'll talk about this a little bit, right? So you can use, for example, principal components. You can use NMDS. So let's keep it at NMDS for now, and we'll talk about what that means, and then we'll try to also do PCA and see how that looks. And now we just need to click on End, Save. And run the pipeline right here. In the meantime, let's complete this pipeline, and let's see what the output would be, right? So in the end, we will get these phylogenies for the data that we have. And we will also have an output of OTU abundance and OTU taxonomy. Now, if we click here, we have some exemplar plots that we can start looking at. So for example, let's go one by one, and let's just see what's inside. So the phyloseq abundance table. And by the way, did everyone manage to create the pipeline and start it and go through all the selection? Just making sure. If you have completed and started the pipeline, just type in yes in chat, so I would know that at least some of you are following along. Anyone? Okay. Yes, thank you. Anyone else? Did anyone else start a pipeline? Thank you. Okay. Yeah, so you're welcome to watch, but if you do want me to repeat any of the steps, or if, as you go through, there are some questions that arise, please go ahead and ask. So again, here we see, right, who remembers what are the 1, 2, and 3? Who remembers what are BKF 1, 2, and 3? Okay, how do we know what is BKF 1, 2, and 3? We can go into our other table, transcripts. No, so these are samples, right? So we have BKF 1, 2, and 3, and these are fecal samples from chow, right? So these are our samples, and these are high-fat diet, right? So we can just color them. These are going to be the chow, and these are going to be the high-fat diet. And now what we're doing is we are comparing them, right? So what would be this number? 411. What would it mean that for OTU underscore 1, we have 411? So this is abundance of this operational taxonomic unit. But if I just go through and I look at all of my OTUs, I have about 300 of these OTUs. You can see that I have a lot of them that have, zeros, and some of them will have pretty small numbers. And then here I have fairly big numbers, right? So as you remember last time, we had a similar situation. We had kind of this table of abundance, right? So this is phyloseq abundance table, the same idea, but instead of individual mapped 16S genes, we have here operational taxonomic units. Now, how do I know what is OTU underscore 1? You will see here that first of all, I have a FASTA file of all of my OTUs. So I can actually get the full sequence of all of my OTUs. I have about 300 of them. All of the 300 sequences are going to be right here. I also have taxonomy table. Let's take a look at this one. So here, for each one of my OTUs, I have that OTU taxonomy, right? So OTU1 is starting from the top, kingdom bacteria, phylum Bacteroidetes, etc., right? And you can see that a lot of them don't have genus and species. And I believe I selected genus. So I will only get information about these ones that do have it assigned. If I was to select family, it looks like I would have a lot more of them annotated, even though some of them don't even have family. But all of them, well, as you can see, 279 doesn't have anything except for the kingdom, right? So now you get the idea, okay, well, how do we go from abundance of OTUs to taxonomy? This is the result of mapping on that database. What else can I get from here? Well, we can see that we have error plots from phyloseq. That is our second file. What are these error plots? So to understand a lot of these outputs, essentially what we have done, right, is we have taken this data to pipeline, and you can look in here, right, and for any one of you that actually wants to implement this on your own workstation or something like that, you can see here the tutorial and documentation, right? Let's see, tutorial. This is, and these are right here, the filtering results, and this is the picture that we are getting, right? So what is it? These are estimated error rates, right? So you can see here that the error rates for each possible transition, these are nucleotide change rates, are shown. And points are the observed error rates for each consensus quality score. So this is essentially a way to look at the sequences of our reads and analyze them for typical errors that arise. Okay, so we see that right here. Now let's go back into our pipeline and see what else do we have. The next one is this file.biome. Okay, so let's open that. Okay, so here essentially we have what's called an annotation file, essentially, that provides all of this information for visualization, because later on we can see what kind of plots are we actually generating. So the plots, okay, let's take a look at the plots here. I chose family, and you see that, you know, some of them are not available. What do I actually see here? Well, I can see down at the bottom, I have my samples, right? So these 3 samples were, what were these samples? These samples were BKF 1, 2, and 3 were chow, and then 13, 14, and 15 were high-fat diet. So these are the chow, and these are the high-fat diet, right? What do I observe just by looking at the family distribution? So for example, in this one, I see a lot more of these specific bacteria, right? I can see, for example, that not all of them actually are that different just by looking at it visually, right? I can see that this one is definitely different from the rest. Also, I see here abundance of different OTUs, and you kind of see how they're different. I see over here that's why we needed to have that TXT file, is to have the library name, and now it's separate right here by OTUs, right? That's the relationship between different OTUs. And this is their abundance. But what's really interesting, right, what we set up there is to have a comparison of diversity. So you can see that this is the alpha diversity measure, and this is Chow 1, and this is Shannon. Now, if you go back into transcriptomics or metagenomics 2, you will read a little more about what each one of these methods is and how they work. But what do we see here? That based on the alpha diversity measure, this one is way up here, right? This one is way down here. And this is kind of like within the range of normal. And you see a similar pattern here under Shannon. However, if you, and this is, by the way, the same thing, but now combined for group. Now, we don't have that many samples, right? And this is something to remember, that really you get statistically significant differences based on more samples in each group. But you can just kind of see how, based on now combining this information together, high-fat diet seems to have a higher alpha diversity. And here also based on Shannon, right? They're over here. This is what's called NMDS. Okay, a question. How is the abundance of one particular bacteria calculated? So again, one particular bacteria, what we did here is based on family, right? So what we are looking at is, how is one family calculated? How is abundance of family calculated? Well, you have reads, and those reads combined together is what would be called depth or coverage, right? So how many reads do I have in this particular segment? The reads are not going to be too short, because the reads are only covering the hypervariable regions, and the hypervariable regions are already short, right? So just because I'm focusing on hypervariable regions, and I know that the hypervariable regions are variable between different families or different species of bacteria, that's how I can calculate the abundance, right? Does that make sense, Marcia? Okay, so, and that's what actually NMDS shows me. NMDS shows me how different are the samples based on all of the data that I provide. And the data that I provide in this example is not the relationship between, not taxonomy like we saw here, but actually this is looking at abundance. So based on the abundance that we have, how can I see the difference between the samples? And you see here that there is a big difference, right? They separate very clearly. This is something like PCA, where we have a dimensionality reduction technique to reduce the number of information points that we have in our data. So we have over 300 OTUs there, and we just kind of compress it to give us variability. How are the samples different between each other based on this information that we have for abundance? And then instead of looking at samples, right, these are samples, I can also compare some of these different families that I have in my data, right? And so here in my data, you see some of these points. Not very clear what I can do with this right now, but just a plot that we can look at. So what did we learn from our analysis results? Well, one, we learned that actually for good measure of alpha diversity, it seems like we need a little more samples for each group, because in the chow diet, they're fairly close together. But the high fat diet definitely shows us conflicting results, right? We have all kinds of things up, down, right? It's difficult to compare because this group in itself has a lot of variability in it. We also know that just by looking at them individually, there are some samples that are very different, right? I would say that this sample and this sample are very different from each other, but they're also both very different from sort of the norm in what we're looking at. So having more data is going to be more important. Having maybe a higher level of taxonomy is going to be easier to compare, right? And also looking at maybe a number of different conditions. For example, I could really combine fecal, cecal, and then chow and diet, right? And if cecal and fecal have the same story, right? What would the story be? For example, the diversity changes, or some representative is much more pronounced, right? That could have better, that would be better information for me to make conclusions about how the microbiota changes affected these mice, right? What kind of stable condition did it create? Now, let's go back really quickly and look. We are looking here at the demo pipeline, right? Let's go see at, take a look at our pipeline. So you can see that it actually finished, right? So it finished here. And I have all of this information in a pipeline that finished. So it's actually a very fast pipeline that you can use. And as I mentioned, here you have a number of different files that you can actually use. You have this full SVL that we had here, right? And in this full SVL, you actually have the fecal and the cecal samples, right? And we can see here, what are their diets? So VKCM 1, 2, and 3. So that is chow. And then we have 13, 14, and 15. So 13, 14, and 15 are high fat diet. And then 1, 2, and 3, and right. So we have actually here a good collection of samples that are representative of both the fecal samples and the cecal samples for both chow and the high fat diet bacteria. Now remember, if you want to run this full data set, or not full, but bigger data set, you will also have to adjust this. So you would actually have to use BKCM 1, 2, and 3, and etc., right? And add that over here with the library name exactly like this is. So this is just for you to remember. Now, let's try and do something so we understand this NMDS, right? Ultimately, how do they compare? We can go and we can actually take this sample or take this information right here and run principal component analysis. So let's try and do that. The way to do this separately, right? We just want to understand better what is the difference and how did we arrive at this result. We can go to unsupervised analysis, and we can just directly upload right here the principal or the abundance for OTUs. So in my downloads, okay, I'm going to take the abundance table, just this file right here. Once it's uploaded, press on continue. And we can use PCA-R library. And what is principal component analysis? Principal component analysis is what we just saw. It shows us the variability between samples based on all the information that we provide. When we just look at the information that we have, the information is rich. There's a lot of features. It has a lot of characteristics, a lot of OTUs, over 300. Difficult to visualize it that way. How do we summarize the data without losing a lot of the data, but really looking at one aspect of the data? The aspect we're looking for is difference in variability, or variability between samples, right? All the samples. So let's click here on yes and press on end. And run pipeline. Okay. And by the way, does anyone have any other questions in the meantime? Because what we're trying to do is I'm just trying to demonstrate how this information could be further analyzed, and then you can have some ideas about, well, how do I actually use these methods to analyze my own data, right? So if I have 2 conditions, 3 conditions, like we just looked at fecal, cecal, high fat down and chow, how do I extract meaningful information out of this data that I can somehow interpret, right? So you can see here that I'm running principal component analysis to analyze variability between samples based on OTU abundance. Then, for example, I can see, well, okay, based on which OTUs. So I can start looking at it from that perspective. Which feature has more significance? Okay, does anyone have any questions about that? And by the way, you can see that in this approach, right, what did we do? We actually took very rich information that had a lot of reads, but we combined those reads in a very efficient way. Instead of mapping each individual read on a particular region in the database, which is a very lengthy, time-consuming process, we're actually combining that information, removing the information that doesn't fit that model, and then we are using that to map on the database. And as a result, the pipeline is much faster, right? So, and that's going to be important. Okay, so here my pipeline has finished. And I can look at the plot and see, well, what did I find in the principal component analysis? So in the principal component analysis, you can see that you have 3 samples on this side, 3 samples on this side, and it gives you several combinations of principal components, PC2 and PC1, PC3 and PC1, and then PC3 and PC2. And the problem with this, of course, is that I kind of see that this is green, but I don't know, is this green or that green? Not very clear. So how can I improve this very quickly and rerun the pipeline to get a better result? The way I can do this is in my table, I just need to add another row that will be called group. And now I can group them and say that all of these are chow, and all of these are high-fat diet. Okay, and now I want to save it. And I'm going to call this annotated. Okay, and it's going to be 1. Just save it as a TXT format. And let's quickly rerun this pipeline. Again, PCA, R library. If you're interested to understand more what principal component is, you can read more about it in our transcriptomics series, where in transcriptomics 2 and 3, we talk about principal component analysis, but also you'll kind of understand how to apply principal component analysis to a variety of different data sets and data types. And it's a useful way to kind of simplify the data, reduce the dimensionality or the complexity of data to just get a sense of what is in my data. Okay, so we're going to give it a couple of minutes to run. We'll see what the output is. In the meantime, let's just kind of review what did we learn. We ran this pipeline that consists of two main portions, the processing, which is the DADA2, and then visualization, which is phyloseq. What we did was we took this DADA2 pipeline, and you can actually get access to this data. It's a package on Bioconductor in R, or you can just build your own pipeline using that tutorial. What this pipeline does is it really helps us reduce the information that we have in raw sequencing reads by sorting all of the data, removing all of the low quality information that we have in the raw data, filtering it, using the the paired end information that we have from the forward and reverse strand, and then organizing the data in these operational taxonomic units that are assigned with taxonomy and phylogeny. And then we create a phyloseq object. You see the biome file is going to be a file that you can use to visualize your data. And then you can move on to diversity analysis. And then we looked at how some of these different outputs look, right? What are they? What kind of information do they have? And you can see in your own pipeline, if you've ran it, what's inside. So inside we saw that we have the sample names. Right here you can see the full table, right? And this is the abundance. This number is the abundance. These are operational taxonomic units, OTUs. And for each OTU, we have the taxonomy. And the taxonomy is really putting them into each group. And we can see that some level of taxonomy is actually not very useful, okay? And some is going to be more complete, more useful, right? So now if you run all of the data together, you can see right here that these are cecal and fecal for chow. These are cecal and fecal for high-fat diet. So visually, you see that they're different, but actually you can see here that with the high-fat diet, it looks like you have more variety, right? And you have some of these. So for example, Bacteroidetes and Bacteroidetes are here, right? These are here. And it's a little bit confusing, I think, just by looking at this, at how do they exactly compare, right? And that's why we need these additional measures to be able to compare them, right? So if we run this separately, two pipelines separately, we can actually see how OTU relationship is and what is their abundance. And then we can calculate diversity either using something like Chow and Shannon or looking at NMDS, PCA, PCOA, etc. Finally, well, let's quickly take a look at what we got in our PCA result, because I think it's finished by now. Let's take a look at this plot. Okay, now we organize them by group, Chow and HFD, high-fat diet, and now you can see, okay, the difference is actually very clear, and in fact, it is very similar to what we see here, right, in MDS. So again, dimensionality reduction method, very similar to PCA. What are the conclusions? How does this correlate with behavior? Well, we saw some of the changes at the microbiome level, but then they did this 14-week period, right, before they took the samples to study their behavior. And so what they looked at was behavioral anxiety tests and basically how standard behavioral patterns compare between the two groups. And you can see that, for example, elevated plus maze, marble burying, those types of comparison gave some statistically significant differences for the high-fat diet. And essentially what it seems to suggest is that there's going to be higher level of anxiety for mice that just had the microbiome implanted into them from donors that lived and that formed that microbiome using on the high-fat diet, right? So some of the suggestion that we have is that while it might not necessarily increase weight, it does suggest that there might be some implication for behavioral and psychological condition that the different diets might have a role in. And I think this is a very interesting result. Of course, you're all welcome to read the publication in full and see all of the different things that they did. How do they set up the experiment? How do they sequence the data? What is it that they did? But pretty much what we did here is very similar to what they did there. You will see how some of the same terminology like alpha and beta diversity, OTUs, some statistical analysis like principal component analysis or principal component analysis or NMDS was used, right? So these different terminologies will now start to make sense as you read the article in full. So I suggest everybody does that if you have the time. Does anyone have any questions about what we covered today? Okay, great. So if no one has any questions, what we'll be talking about next time are metagenomic projects. So we're going to start transitioning from how we analyze data, what is the data, how is the data prepared, to what types of applications. So far we saw two. The first one was really a practice for us to just kind of see what's in the data, right? We looked at the human microbiome American gut project, and then we looked at the mice and the relationship to behavior. But we learned in the beginning that metagenomics actually covers a variety of different type of genomic information, from viruses to bacterial resistance, to rhizobacteria in plants, to the microbiome in human gut, right, or animal gut. So we see a lot of different applications of the study of microbial genomes. We'll talk about some of those examples again soon. And then next time we'll just talk about, metagenomics projects. Just start with an overview, different types of data, where to find this kind of data, how to understand which pipeline would be most useful for what type of analysis that you want to perform. So again, thank you everyone for joining this session today. I look forward to seeing you again next week. And I invite all of you to stay in touch with us and ask us any questions that you have about the material that we covered today, or about your own idea for your own project as you start thinking about how to develop a project of your own. If you don't have any more questions, this will be it for today. And again, I'm excited to see you again next week.